chapter 3 of my dissertation, which is understanding the correlation between environmental morphology and the subfamily Cassidinae. So the outline of my presentation includes giving a brief introduction to what ecogeographical rules are, how does it apply to the group that I'm working with, and how I tested for it. So environmental gradients, they are in correlations with morphology. They are very important because it helps understanding the diversification of groups. Among these environmental gradients, these correlations of morphology and environmental gradients, we have several that, I'm sorry, let me back up a little bit. So they are known as ecogeographical rules, and there are many ecogeographical rules. Sorry. Was I supposed to?
Yeah, we started technically.
Should I start again?
If you'd like.
Oh good, I have more time then.
Sure.
So among the ecogeographical rules, there is Bergamon's rules, Gobler's rules, and these are just a few of them that there are out there, trying to correlate temperature, precipitation, humidity with morphological features. So the subfamily Cassidinae is the largest subfamily, the second largest subfamily of leaf beetles, with 6,000, almost 7,000 species worldwide distributed. It's a monophyletic group, and currently 12 tribes are known for it. Among them, we have the Durinotine tribe, which is this really cute tribe that I work with. They are characterized by the presence of this very conspicuous spine in the back of the elytra. They have 5 genera, around 64 species, and they have neotropical distribution. But when you look at the distribution and you put them together, you see that there's a sort of latitudinal gradient, that latitudinal climb in the morphology, where the ones further up in the neotropics, closer to the equator, they don't present the spine, or they just present a small tubercle or nothing. And the ones further south, they present this very large thin spine. So the question is, and when people look at this, many researchers have brought this up, what actually leads to that. In 1923, Franz Bayet, which was like a huge researcher in this family, he suggested that there was an actual correlation between the environment and morphology, where in different pockets of neotropics you would have the presence or not of the spine. And actually the spine would enable the invasion of cooler areas of the tropics. So, yeah, and so the goal of my study is actually to understand, to test what Bayet suggested, whether the presence or not actually it is correlated with the distribution, and also if the size of the spine matters, because some of them, the species that are further south, you can see that there is a differentiation in the size of the spine. So I wonder if the size actually matters. For that, I used the hypervolume package, which was put forward by Blonder in 2014, and it's a package in R that uses the concept of niche proposed by Hutchinson, which describes the niche as the set of environments where the species can live. So using this concept, you can take the set of environments and the set of morphology that defines the group, and creates hypervolumes that would actually describe the species. So in the environmental hypervolume part, I used environment values associated with occurrence points. And in the morphological hypervolume, I use morphological traits, measurements of morphological traits. And the methods consist basically on estimating these two hypervolumes, estimating the overlap, which are like kernel densities or calculations of overlap. Then with the matrix of these two factors, I then test for the correlation between them using the mental test. So for that, I tested in genus and species level. Genus to understand whether or not the presence of the spine would make a difference in the distribution, And in the species level, to understand whether or not the size would matter, I use only the species that actually have the spine. This graphic just shows the species. In orange, it shows the species that were not included in the experiment, because in the testing, because many species in invertebrates, as most of you know, they are only known from one species, and some of them are very damaged. So I couldn't take all the measurements, and you can't create a hypervolume when you have one of the traits not measured. So you have a hole basically. Then you can't compare them. So in the genus level, I use 4 genera because one of the genus is monotypic, is only known from the species type, the type species, and this type species is very, very damaged. And in the species level, I use 16 out of 22 species. The morphological traits include pronotal length, width, elytra, width in the humeral level and in the spine level, and elytral length, and the spine height on lateral side, lateral view. The occurrence data, well, I got most of my occurrence data from material from collections. Like, I visit more than 20 collections, among them like institutions in Europe, North America, and South America. I did also field work, so material collected by me, and some data from the literature. And some of them didn't have georeference, so I used Google Gazetteer and Google Earth to get the coordinates for them. In the environmental data, I used world claim layers. I excluded 4 of them because they were very correlated. Bio 8, 9, and 18, 19, which is like precipitation of wettest and driest month, and temperature of coldest and hottest month. So I took them out, and I used the PCs, the principal component. I ran a PCA because to reduce like the redundancy of the data also. So basically, through the whole project, I use principal components, not only for environmental data, but also for morphology. So this slide summarizes basically the methods I use in order to increase the sample size, the occurrence points that I had for the species. I use niche modeling techniques. So I use MaxSense to estimate the potential distribution of most of the species. I threshold this distribution and a random sample within it. And this random sample of points were used to extract values from the 3 first principal component layers that summarizes the neotropical area. They explain like 20 and 90% of the environment. And in morphology, I use also all the principal components. Then I use the hypervolume package. I estimate the volumes of each species and each genera. I created the overlap matrices, and I used the mental test to check for correlation between them. As a result, in the environmental hypervolume, I could notice there was like very, very high levels of overlap, which shows that the neotropical area is very uniform. And you can see in this table on the right bottom side that they actually, between different genera, there is a high level of overlap. The same thing with species, and in the left side you can see this is a pairwise matrix showing different hypervolumes of different species. And you can see also that the levels are pretty high. On the contrary, in the morphological hypervolume, there were very low levels of overlap, which shows that there is like a great morphological disparity among genera. And you can see distinct groups in this image. Different colors are very separated. And in the species level, although you can see that they are actually very overlapped in the image, when you look at the table, they have very low levels of overlap with spine and without spine, which shows that actually having spine or not doesn't represent any kind of difference in their overlap in the hypervolume. So after calculating this overlap matrices, I use the mental test, and you can see in this table that the mental test shows that there is no significance between the correlation of environment and morphology. In the first column, principal components always means that I used all the principal components in the morphological traits. In the second one, I took the PC1 out because it was representing like 83% of the variation. So the variance, so I just thought, well, maybe this is influenced somehow, but actually that didn't. And then in PC2, 3, and 4, I just tried to make an equivalent comparison with the environment hypervolume, which had only 3 PCs. So, but it actually didn't make a difference either. So, as conclusion, the suspected Cline in the group doesn't actually truly exist, and it's probably an artifact. Currently I'm working on the biogeography of the group, and it seems that actually vicariant events that actually drove the diversification in the morphology and distribution of the group. This is one of the best points of conclusions of this work, is that actually the morphological traits that I picked to represent those groups were pretty reasonable, and they actually were able to discern different genera and different species. And the main conclusion of this work is that, well, hypervolume package is very useful, and using it along with other techniques can be very interesting to understand correlations and other relevant biogeographical and ecological research problems. With that, I end my presentation, and I would be glad to take any questions. Yes?
Does spine size show any relationship with the body size of?
No, it does not. Yeah, I couldn't find any kind of correlation with other measurements either, and that's one of the reasons. I mean, other measurements like elytral length and width, sometimes they might be correlated. So that's why I use principal components for the morphological traits, just to eliminate this problem.
Did you check the environmental differences among the places with the spines, but just maybe purely kind
I'm sorry, can you repeat?
just maybe a relationship of the spine with latitude?
No, the thing is that to calculate the hypervolume, you can't have only one trait. You need to have different traits because you need a dimensional figure of that. It's fine. So that's I didn't test only with spine because I couldn't. But I mean, I added this spine in the data, and I took it out, and it didn't make any difference. So I assume that doesn't, like, the spine is not correlated with any part of any different in the environmental hypervolumes along the latitude of the topics.
So are there any good explanations for why that spine exists?
Well, I assume that these are just vicariant events that happened, and eventually, because the most derived ones, they actually present this